Hi, here we will discuss about the DNA. What is the DNA? So basically we will understand. So here is the human which that is you can see. And you can say that is the animal and the plant which that contain all cells is the somatic cell is contain nucleus inside, and the nucleus contain chromatin material in the form of chromosome. During the cell division it will be appear during the condensed formation, and bacteria and fungi is also contain cells, so it means the nucleus. Even all microorganism and macroorganism which contain a nucleus in the cell. Each cell contain a same nucleus which contain here is a chromosome. This chromosome is made up of chromatin material, and this is attached with the centromere, and that is the in the form of pair, and that pair is the half come from the mother and half is come from the father. So the chromosome contain basically the 40% DNA and 60% protein. 46 in somatic cell discovered German embryologist Walter Fleming in 1882. So here is the histone protein which that is contain basically is the 60% is contain H2A and H4 and H2A and H2B, which that is a four type of histone protein contain a 8 subunit, and this is the basically used for the condense and attachment of the due to the positive charge, and the DNA contain negative charge, so the attract to each other to bind. So the important thing is the lysine is contained, the lysine rich histone protein. So here is the histone protein which that is wrapping around the DNA is wrapping around the histone protein. Here is the DNA which contain nitrogenous base, adenine, thymine, guanine and cytosine. Adenine always we contain the thymine and guanine with cytosine always. So here I am drawing that is the type of chromosome in the different organism contain a different type of chromosome. For example, telocentric. Telocentric means the spindle fiber are present and the centromere is the side wise. While the acrocentric means the little bit chromatin material are present in the upper region, while the down region is more. And the metacentric means the between the chromosome the centromere is present, while the submetacentric is the a little bit upper part. So let's begin to understand the chromosome in the form of somatic cells. So here I am drawing just one chromosome for understanding. So this is the loci, and this is the neighboring gene of the one chromosome. So this is the homologous chromosome to each other. So in this way here is a come from female and male. Half is come from male and half is come from female. So the capital T is the dominant, while the heterozygous alleles, and there is the different genes here, and here is the homozygous dominant allele, and here is the homozygous recessive allele. It means the recessive, it means not show the phenotype, while the dominant can be show the phenotype, but it can be heterozygous or homozygous, because the one is dominant and one is recessive, or both are dominant trait. So here is the what is DNA, just quick overview will discuss. Deoxyribonucleic acid is a molecule composed of two polynucleotide chains that coil around each other to form a double helix helical structure, and carrying the genetic instruction for development, functioning, growth and reproduction of all known organisms and many viruses. And DNA and ribonucleic acid or nucleic acid, basically it is the acid and contain a negative charge, so that is why the protein contain a positive charge, histone protein, with lysine rich content amino acid which that attract the DNA together to bind. So that is important things. And on the other hand, the methyl group and acetyl group can be added to show the epigenetic. So here is the structure, is the diameter of the DNA is the 2 nanometer, and here is the anti-parallel 5 prime to 3 prime one strand, and the other is the anti-parallel 3 to 5 prime. So the backbone contain a ribose and phosphate group, While here is the nitrogenous base distance is the 0.34 nanometer, while the 1 is the minor groove and major groove contain a 3.4 nanometer diameter. So let's quick overview about the content with the biochemical formulas. So here is the deoxyribose. It means here is the only hydrogen. So this is the DNA only, present in the DNA only. While here is the ribose. This contain a hydroxyl group. So that is always present in the RNA because ribonucleic acid. While the deoxyribonucleic acid means the deoxides. So it means the oxygen are not present. Here is the phosphate group. And on the other hand, the purines is the double ring nitrogenous base, adenine and guanine. This you can see in this diagram. While the pyrimidine is contain a cytosine, single ring, and uracil. Basically, the uracil always contain the RNA, not DNA, and thymine. Thymine and cytosine is a part of the DNA, while the uracil are present with the alternate of thymine. So where the thymine present, and here is the adenine with thymine contain double bond, and guanine with cytosine contain a triple bond. So let's begin to understand. Here is the binding, and we will discuss about the strand of DNA. So here is the phosphate group, and here is the ribose sugar. Here is the downward 3' to 5' movement. It means the 3' to 5' movement, and this is the phosphate group attachment. And in this way, here is the phosphodiester bond. This phosphodiester bond are present. This phosphodiester bond is contain a diester, two ester bond. So that is why it is known as a phosphodiester. So it contain a 5 carbon one side, and the one side contain a 3 carbon. So it move from 3' to 5', and the other strand will move the 3' to 5' is anti-parallel. So remember about these things. So let's begin to understand the chromosome is here, and again we will discuss about the another diagram to understand the DNA structure. So this is the DNA double helical structure you can see in this diagram. So here is the 3' end in one side, while the other side is contain 5' end of the carbon, which that's contain phosphate group in the backbone of the basically is the basic strand, which that inner side is the nitrogenous bases, which that is bind with the hydrogen bond. So the thymine always bind with the adenine, and guanine is always bind with the cytosine. So the guanine bind with the cytosine with triple bond, and the double bond alternatively the double and triple bond will give the double helical structure because more and less hydrogen bonding. On the other hand, here is let's begin to understand what is the mechanism and what is the function and the control our body with the phenotype. And what is the phenotype? For example, here is the tissue during the developmental stages to produce the a zygote to produce the liver, muscle, stomach, and bone, which that is due to the epigenetics, the DNA methylation and demethylation. So that is why demethyltransferase enzyme used for the and acetyl transferase enzyme used to function the activation of the gene for the phenotype for the production of the content or material in the cell for proliferation and changes of the zygote into the another the angiogenesis and the osteogenesis, etc. So that is why here I am drawing the cell which that's contain. Here is the plasma membrane, extracellular space, and here is the cytoplasm. When bind the growth factor or ligand with the tyrosine kinase receptor, after the binding, so the phosphorylation in the intracellular domain of the tyrosine kinase receptor to trigger the phosphorylation of the another protein to the chain reaction, and this will be triggered by the kinase enzyme used to become a transcriptional factor. This transcriptional factor will move into the nucleus. So here is the nucleus contain DNA. in the form of chromosome, and where the requirement of the gene activation, so the acetyl group will be added. So remember, after the binding with the promoter region to express the specific gene activation, so this promoter region will be activated due to the transcriptional factor, and in this way the RNA production will be occur, the RNA promoter region, which that produce the RNA through a transcription, and the polymerase enzyme used for it. And after this, the messenger RNA will be produced, and this modification by the exon and intron splicing mechanism to move outside, and where the uh, rough endoplasmic reticulum used to ribosome bind and transfer RNA used to decoding of the messenger RNA to produce the protein is known as translation. And this protein is used for the in the form of vesicle is used intracellularly and extracellularly for the exocytosis. So that is the thanks for watching. That was the DNA. So I hope you are covering this concept about the DNA. So please make sure to subscribe, like, and share. Bye.